a real pleasure to be here. Thank you for inviting me. So this is a picture of a female Anopheles mosquito, and there are over 200 million new cases of malaria across the world every year. And the one thing that each of those people who catches malaria has in common is that they have each been bitten by one of these who is carrying the Plasmodium parasite that causes malaria. And it's this reliance of the parasite on the mosquito to get from person to person that has made vector control, so mosquito control, the mainstay of malaria control. It really has made the biggest difference in terms of controlling malaria, to actually target the mosquitoes just as much as the parasites. So this just gives a picture of the situation globally. You can see that the hotbed of malaria in terms of the deaths each year remains in Africa. And it's disproportionately affecting children under the age of 5. This is the blue depth here. You can see the vast majority of children who are people who are dying are children. So there's two major ways of currently controlling mosquitoes in Africa. One is called indoor residual spraying, where people go in and spray insecticides on the walls of the houses where people live. And the other is insecticide-treated bed nets, here on the right. And these two ways of controlling mosquitoes are super effective. But the problem is that mosquitoes rapidly evolve insecticide resistance. So the bed nets still offer some protection because people sleeping under them, even if there's no insecticide delivered. So what happens is the mosquito tries to probe, tries to get under the net, and the net is covered with insecticides, and the mosquito is given a dose through that contact. And on the walls, the same thing. Actually, the walls are, in a sense, people are bitten, and then the mosquito gets the insecticide because it goes to rest on the wall. They take in a blood meal that weighs more than themselves, so they go and rest and digest on the wall and get the insecticide dose there. But in that top map, everywhere where you see that dark brown, that means that mosquitoes who are transmitting human malaria have evolved resistance to all the classes of insecticides that are currently available. So in our efforts to try to intervene, so we want to do malaria control, we want to intervene, we want to push mosquitoes down the path to eradication, push malaria down the path to eradication, but we keep on pushing them down the path to resistance. How can we do better? New interventions are coming. There's novel insecticides in the pipeline, and I think in a particularly exciting, which I used to be extremely skeptical about, but now actually I think technically it's solved because of CRISPR, but it now is just a political problem about a genetically modified release of mosquitoes. I think gene-drive release will happen in the next 5 years, and we need to be in a position to understand how is this going to sweep through populations in Africa, and how are mosquitoes evolving resistance so we can get ahead of the resistance and not keeping falling down the same path. So the group that I have at Sanger, we really conceive of the future as a place that we want to do the kind of research that allows us to have a much better chance at really harnessing the power of vector control and doing it in a strategic way where we are ahead of the evolution of resistance. So to understand how alleles, the genetic variation that sort of underpin resistance, spread through populations, we need to understand how those populations of the mosquitoes that do the transmission are connected in space and time. So can you have a population where you release gene drive in Burkina Faso and see it spread all the way to South Africa? Or are there cryptic species in clusters that means that actually that effort will be blocked and you need to do release in a much more targeted and strategic way? Understanding something about the genetic connectedness of populations is really going to help us understand how to deliver gene drive more effectively and strategically. And it also, we've been able to kind of learn a little bit about this through the use of insecticides because we can see over time the resistance alleles spreading through populations. We know kind of molecularly what underpins resistance, and we can watch those mutations move over space and time, and we can see where they move, and we can also see where they don't move, even though there's pressure. So most of what we do actually really heavily relies on having reference genomes. And I will talk to you in a minute about what I think of as a reference genome. having it be a high quality reference genome is really important because it allows you to see all the genetic variation in an individual organism. So effectively, a reference genome is like the thing that you map to. So if I was to sequence everyone in the room, I wouldn't have a reference genome for each of you. I would actually just sequence you with a short read, simple, cheap, effective tool, and then I would map your DNA to the human reference genome. And I would understand how each of you is different. We're all different in a very small number of ways. 99.9% of our A's, C's, T's, and G's are actually identical. But mosquitoes have a lot more variation. So when we have a high quality reference genome and we do resequencing, we can understand the genetic variation in a population. We can understand how evolvable those species are and those populations are. We can estimate population size based on the number of variants that we can identify. We can also see the structure. That's the thing I was talking about earlier. How isolated are different populations? How much are they sharing alleles? And this is really also very important for doing gene drive design. You have to be able to find the places in the genome where actually you can target and there's not a lot of genetic variation already out there as resistance. So what's a reference genome? It's basically one instance of each chromosome of an organism. So we have 23 pairs of chromosomes. Building that first reference genome is akin to trying to put together a jigsaw puzzle when you don't have the picture on the box. So you see the pieces, but you have actually no idea how the pieces go together because you don't have a reference to look at. And of course, this is much easier with big pieces, which is why actually reference genome sequencing has taken off in the last 5 years in a massive way, because long reads have made it so much easier to put together genomes. So when you have these big pieces, instead of all the tiny little pieces from Illumina, or even Sanger sequencing to some extent, which was around 700 nucleotides instead of 200, it's much easier to see how they all kind of line up and go together to build those full chromosomes in genomes. So the quality of this final product, this is the reference genome now, the puzzle pieces have put it together, really matters. If we have a reference genome that is missing the antennae or the duck here, every time we look at additional organisms mapped to that, we will never see those pieces of DNA because they're not in the reference. So that high quality is really important. So building high quality genomes from single insects started actually just 6 years ago. We were the first ones to do it with a mosquito, using a single mosquito. And this is completely the norm now. We've sequenced the reference genomes of over 1,000 insects now, just using one single specimen. It used to be that you had to grind up buckets of insects to get enough DNA to generate a reference genome. And that caused a lot of problems, especially for insects, because they're so genetically variable. So when you see a base when you see a different mutation every 50 or 100 bases, and you're trying to put them all together, it just becomes it becomes an impossible task, basically. If I have time, I will at the end talk about a project that we're doing at the Sanger Institute called the Darwin Tree of Life Project, which you may have heard about. It's an effort in the UK led by Sanger, but actually partnered with many biodiversity organizations across the country to create high quality reference genomes for all the described eukaryotic species found in the UK. And we've done 2,500 already. Okay, so this is the kind of continental distribution of the 4 worst or best vectors of malaria in Africa. Anopheles gambiae, Coluzi, and arabiensis, over the last decade, we've sequenced thousands of their genomes, and we really understand a lot about their population structure. They are sibling species, so they actually can interbreed if they want to. They tend to be pretty good species in the sense that they don't crossmate, but what that can mean is that if they do crossmate, those alleles can move between species as well as between populations. Today I'm going to talk mostly about funestus, which is another major vector, and actually it's longer lived than Anopheles gambiae complex, and it's thought to contribute more heavily to malaria transmission in the places where it is the dominant vector. Briefly, I'm going to give you this glimpse into Anopheles gambiae and Coluzi. And this story may seem a little familiar I just heard Henry North was here recently, if you saw that talk. It's a very similar concept of kind of allelic movement through populations. So this picture of this tree here is looking at one very important gene in the mosquito genome called the voltage-gated sodium channel. It's a mutation at one amino acid in that gene confers DDT resistance and pyrethroid resistance. So it's been under very strong selection over the course of the last 60 or 70 years. that same amino acid, so those same 3 bases that code for that amino acid, it can turn into 2 different amino acids by a different mutation at that 3 bases that confers resistance. So it's 2 convergent mutations. And for a long time, people knew about this and thought actually there's a phenylalanine in the east of Africa and a serine in the west of Africa. And these things arose once and have swept through these regions of Africa. And so this was the lore for a very long time. And now that we've been able to sequence beyond, so not just that amino acid to say this is a resistant mosquito or not a resistant mosquito, we've sequenced the linked DNA attached to each of those and what's called the haplotype. We can look at the full view of actually how genetically different is each of these haplotypes that confers resistance. And what we've identified is that there's at least 10 different times that these mutations have happened on different genetic backgrounds. So it's a huge amount of convergent evolution. Effectively, these sweeps are happening all over the place independently. And sometimes they happen and they can actually migrate huge distances. So although they're happening locally, the countries are colored in different colors here. So you can see that there's there's a nice green cluster here and a blue cluster here. Actually, in this one, it's red and blue intermingled. And that's because it's not only swept a very large distance, but it's also crossed species boundaries. So when the pressure is strong enough, alleles will move through space and through species. This is the group of folks in my team who have been leading this Anopheles funestus, so I'm going back to the other mosquito now, analysis that I'm going to talk about. About maybe 6 years ago, we put out a call to our colleagues in Africa who are vector biologists, and we said, if you're interested in participating in a study to get a handle on Anopheles funestus population structure, and you're out there collecting any specimens, would you be interested in partnering with us on this project? And so people from across Africa collected funestus and sent them to us, and we sequenced them. This is the Anopheles genome has 3 chromosomes. So we have 23, but they have 3. And those big yellow kind of switch things here are inversions. Anopheles mosquitoes are famous for inversions. They have a lot of segregating inversions where a whole giant chunk, 10 million bases or 20 million bases, is flipped in its orientation. And these often harbor adaptive genetic variation. So you can see different inversions changing over clinal gradients, for example, through space and through time with aridity and with different, as you go from the forest into the savanna. So this reference genome that existed for funestus initially is on the top here. And you can see all these little tiny black dots. That's everywhere that the reference genome was broken. We were missing some DNA and we don't know what it was. With this new approach, with the improvement of long-read sequencing technology in the last 5 years, we created a reference genome from a single mosquito that was wild-caught from Gabon, and you can see there's just a few black dots, a few places where we don't know what's in there. We can't put it back together, and it's probably a real high density of repeats that's very difficult to sequence through. So this is a principal component analysis looking at the one of the arms of the chromosomes. So this is looking at 2L. And we look at 2L because actually these little things that look like inversions are actually errors in the other reference. They're not actually real segregating inversions in the population. So this is an arm that doesn't have a lot of genetic structural variance. So we look at that arm and the SNPs, the single nucleotide polymorphisms, when it's an A instead of a T, all of those on that arm. We downsample to 1 million of these biallelic variants, and we then create this dimensionality reduction view of what is population structure in this species. And what you can see here is there's this one red circle here, and that is this same red circle right here. So across the entire equator, these populations are genetically connected. Whereas right here, this little triangle, green triangle, and right here of North Ghana, and this little cluster here in South Benin, that's here, Benin and North Ghana. They're genetically distinct from this equatorial population, but you would never know it to look at them. They all look exactly the same. And the only way to know this is by sequencing their DNA. North Ghana looks to be actually a population that's gone through bottlenecks, perhaps because it's on the north edge of the population range or because it is under serious control. And so actually the populations have crashed and recovered. We measure this by looking at what's called runs of homozygosity. So this is, you can see here, this is a picture of the genetic variation plotted across a chromosome. And you can see this big stretch here where there's no genetic variation. That means that the allele that individual inherited from mother and from father is genetically exactly the same, which means somewhere recently they share the same, there was inbreeding basically, they share the same ancestry. And you can see lots of small tracks of that too, all these little tiny dips where there's runs of homozygosity. That means longer back ago in time, there was inbreeding. And now that's had a chance to erode a little bit. It originally looked like this, but through recombination bringing in new variation, those spots have eroded. Whereas in a neighboring population in Central African Republic, you don't see any of these except for one place right here, and that is a selective sweep. That dip in heterozygosity that we see it in every single population that we look at because it is the resistance to pyrethroid locus, full of cytochrome P450s that confer resistance. And that story is exactly the same as the VGSC story I just told you about from Gambi, where actually these events have happened in convergence. So every time there's a resistance mutation, it's arisen independently and swept locally. So this is just a view on that equatorial population. There is structure there. The country colors are different here. However, compared to these other populations that are even really nearby, the structure is much less obvious. This is looking at these inversions, and this is the same PCA, and this is why we didn't do it on the other arms, because you can see how complicated it gets thanks to segregating inversions. But we've got a new way of looking at this on the genome instead of in these PCAs, and I really love this way of looking at the data. So now we're looking at the X chromosome, and each of these three colors that you can see here, this is actually many colors, but they're overplotted, so it looks a bit purple. You can see that each line is an individual mosquito, and we're looking at its PC1. So if you look at this plot here, PC1 is on the x-axis. So here you can see that those equatorial populations are quite different from those southeastern populations on PC1. PC1 is separating those two populations. Now we basically flip that PC1 and put it onto the y-axis. And instead of doing it for the whole arm at once, we do it in windows along the genome. And what we can see is the structure in each of the places along the genome. So X chromosome is really boring. The southeastern cohorts, they just kind of tick along. They're basically always consistently different from the equatorial cohorts. And there's some interesting stuff happening here, but it's not very precise. You're sort of seeing a big window, and it's not giving you a great sense that there's something really structurally different there. These kind of are nearby, and they come together a little bit closer. Now, if we look at so this is the same populations. This is all the populations here. But now we're just looking at two populations because it gets so confusing otherwise. So turquoise is Mozambique, and Cameroon is in red. And we're looking at chromosome 3 instead of X. You can see so much more structure here. And what the structure is identifying is those inversions. So if you follow each line along, you can figure out the karyotype, which inversion version that individual is carrying. So along here, it might be 3RA, A, so it's got both copies of A, but this individual here is heterozygous for it. So it's got one wild type and one A inversion. And the same thing here. 3LA is very different because you can see there's this kind of coming together again in the middle. And we believe that actually Cameroon has no variation. So Cameroon is fixed for 3LA. It doesn't segregate it. All the individuals don't have the inversion, whereas these individuals do. And so what's coming together here is actually probably, we think, it's a large inversion. And if you recombine in the middle of inversion, it's lethal because you're missing a chunk of the genome. But if you double recombine within an inversion, you actually can recover from that event. And so double recombination events are rare, but they do happen. And they're happening enough here that we think that's what's happening in the middle of this inversion, is double recombination has eroded the signature that really differentiates the two different versions of that karyotype. And interestingly, when we look at our actual individual mosquitoes, we can see that some of them actually take a path where they jump back up and then jump back down. So this individual, it inherited 3RB from mother and from father, but one of those parents, back ago in its ancestry, one of its parents must have recombined, double recombined, and it took a piece of 3R plus. So that piece is still sitting there in this particular individual. When we look at the whole genome, so this is the same view now, but with all the populations and all the chromosomes, here's the X that was kind of boring, and here's all the other arms. The places that really pop out besides the inversions visually are actually all known insecticide resistance loci. And that just goes to show the power of the control that we are trying to put onto these mosquitoes, how much we're having an impact on their genomes. This is a fascinating story. So this is a way of looking at selective sweeps. And this is that locus I was saying to you, RP1, resistance to pyrethroids, that every single population has a sweep there. You can see they all pop up there. That basically means that every individual in that population is sharing the same allele. They look like that run of homozygosity because there's been such strong selection, there's not very much variation. The mutation that confers resistance has swept. But each of these populations is doing it differently. And it's actually a lot of copy number variation that's happening there. I won't really talk about that today. I'll talk about the gene GABA, but this particular story is too interesting to not mention. VGSC, the picture I showed you from Anopheles gambiae, where there's been 10 independent selective sweeps. For some reason, that mutation, even though it's present in many insect species around the world, and it's always responding to DDT and pyrethroids, actually has never been seen in Anopheles funestus, and nobody knows why. This is a widespread continental species, completely exposed to pyrethroids and DDT, but for some reason has never swept this particular mutation that is known to confer resistance. And it's been a bit of a mystery. So it must be some kind of cost that is preventing it from sweeping. There must be a cost to having that mutation that confers resistance. So we were really bewildered when we found that there was some small population in Tanzania that actually does have that mutation, and it is swept, every individual. We hardly sequenced any from this population. I think there were about 10 individuals. None of them across the rest of Africa have any sign of that mutation, and all 10 in that one population have it. This was actually a really fascinating phone call. We were on a Zoom call discussing this, and somebody just Googled Morogoro and DDT. And it turns out that Morogoro, which is the region in Tanzania that these samples are from, has a DDT stockpile that has been leaking DDT into the environment for decades. And it's only just now being removed. These are pictures from the actual warehouse where the barrels of DDT have been stored for decades. And so I imagine that everything in that region has been exposed to extremely strong pressure and has, if they've survived, they have DDT resistance. Okay, so how do we understand when these mutations arose? We've been really interested in trying to go back in time and understand a little bit more about what did population structure look like before people were using insecticides. And that has to take us back pretty far. Before the 1950s is when the DDT started being used widely across Africa. So we have a project that we call affectionately Neandersquito, because if you go back 100 years ago to a pinned insect, it's basically the same amount of time as going back to our Neanderthal populations. One of the other fun aspects of this project, besides being able to kind of go to museums and try to find their historic collections, is there's a great archive at the London School. And so the postdoc who was working on this managed to go and dig into all the books and papers and notes of Herbert Leeson, who traveled by plane from Croydon to Entebbe, Uganda. I have a couple pictures of that in a minute. And kept all of his papers, and they're at the London School now. And you can see some fun stuff, like there were glazed sheep tongues on the lunch menu on the train. So this is there's a really wonderful, actually, like maybe 20-minute film footage of this event. So you can see him flying. It took 5 days to fly from Croydon. They're flying very low across Africa. And there's the film was from, I actually don't know what year it was made, but probably soon after 1936 when they were there. And he took with him a assistant called David Gillett, who's actually narrates the film, I think in the 1980s or 1990s, many years later. So this is a picture of the car that they drove around Uganda in 1936, and this is Leeson bent over a microscope. You can't see it very well in this picture, but you can see it quite well in the film, looking at mosquitoes under the microscope. And there's some letters in this that we just enjoyed going through these materials, trying to kind of promote. As we know, we have our colleagues that we want to give a leg up in the world, and his scholastic record was so bad, but in every other respect, he is a very unusual and able person, one of those born naturalists, and to speak snobbishly, a gentleman. A very long letter of reference for David Gillett. Here's, in the film footage, there's somebody using a ladle to scoop the larvae out, and I have to say this is still a essential bit of vector biology equipment. And we really identified with this confusion because these mosquitoes are so difficult. You cannot see morphological differences. They all look exactly the same. And they're trying to figure out by the wing patterning which mosquito is which. And they're just frustrated and basically put Anopheles X because they have no idea. This is us in Uganda in 2016. That's my daughter, who's now 14. And I just wanted to bring this up because we're still using the soup ladle to collect mosquitoes. Okay, so where do we get all these historic mosquitoes? Museums have huge collections, and Leeson's collection was in the Natural History Museum in London. And so we sequenced Leeson's mosquitoes as part of this project. You can see here, it's a little bit tricky to say, but I think that says like 1912 or 1916. These are old, and this mosquito in particular, she still has a blood meal in there. So not only do we get the sequence from the mosquito, but we can also see if the mosquito bit a human, we can see the human DNA, and we can see the parasite DNA if it's present. So we had to get permission to sequence these pinned insects because the Natural History Museum does not want you to grind up their insects. And so until this project, when I went to the Natural History Museum to pitch this project, they were mortified at the idea that I would be grinding up the mosquitoes. And I said, what if we just took a leg? No, absolutely not. You can only take the specimens if we get them back as good as we gave them to you. Which is not a small task, considering that for all of our modern mosquitoes, which are full of DNA, not subject to decades in an insect collection, we grind them up. So we developed an approach where you actually can leave the mosquito on its pin, dip it into a lysis buffer, and kind of leach the DNA out. And this DNA is ancient, well, historic. It's in very small fragments, typically 30 to 50 bases, instead of the nice long fragments we get from a modern mosquito. And it's got signals of ancient DNA damage caused by water over time. This is a kind of schematic of the approach. So we first, because they're very dry and brittle and subject to static electricity, so we first put them in a box with some moisture in the box, some humidity, and then we dip them in the lysis buffer and we take out their DNA. And then we have to give them back to the museum. We put them through something called critical point drying, which we call refluffing, because the antennae get all kind of warped and bad looking, and then they go right back into looking very nice. And sometimes they look better than they did when we got them. So when we looked at their genomes from the 1930s to the 1970s, we could see that populations colored here, so the black dots are now the historic genomes. Based on where they came from, they still actually looked very much like what the modern populations look like. And we could see that none of those insects, so the black line now here is the historic mosquitoes. And you can see in these places where there's big insecticide resistance, so big control efforts changing the landscape of the genomes of these organisms, you can see no black lines following that path, except in this particular region. And this is GABA. This is a gene that confers resistance to a different insecticide called dieldrin, which was in brief usage in West Africa in the 1960s, and resistance evolved super fast, and so they removed it. But it's also a long-lasting insecticide like DDT, so it stays in the environment. They knew, based on the phenotype, that the resistance was there in the 1960s in West Africa. They did not know the mutation at that point, but later on it was discovered. And we can see that actually, although dieldrin is not in use, this is the same kind of picture showing you that everybody in Cameroon and Ghana and I've forgotten, Nigeria, basically has no genetic variation in this region. This is like, they're all sharing that same haplotype. So there is still very strong resistance happening in parts of Africa that on this locus. And it's a little bit perplexing as to whether this is some story like the DDT story of Morogoro, where there's dieldrin still in the environment leaking, or whether there is some actual benefit of these mutations to other insecticides that are in use now. So these were these 6 individuals that followed this GABA path. And then we looked at the actual mutation, and you can see that the modern resistance are green, and the historic, the historic resistance have a little bit of green there. So it is that same actual amino acid mutation that is present in those historic samples. We wanted to know if it was the same haplotype. So is that same long region, was that the thing that is now still present in our modern samples? But it's been very difficult to disentangle because there's so much genetic variation here. You can see this is just 9,000 bases, so 9,000 A's, C's, T's, and G's. And every place there's a color means that it's a variant compared to the reference genome. So there's a huge amount of variation. If this was a human, you would see like 5 of those color bits. So implications. population structure is driven both by geography and by unpredictable factors. So we can see a latitudinal gradient, but we can also see things like Benin and North Ghana, where we don't have explanations as to why they're genetically different from the rest of the populations nearby. We also think that it has remained stable for the last 1,000 generations, which is really important to know for things like gene drive, because actually that means that this isn't just a fluke. We should be thinking about targeted release of gene drive in East Africa and West Africa and Central Africa separately, because those structures are stable over time. We think we need a mind shift in how we approach resistance. We have to be assuming that resistance is out there already, not that it will eventually evolve. And if we approach it that way, then really what we need to be set up to do is to monitor for resistance. We need to know where in the genome to look, and we need to have the right tools to do it. And whole genome sequencing is wonderful for being able to identify sweeps and seeing pressure come through, the fingerprint, the footprint of selection. But actually you have to sequence a lot of genomes to get that sense, and you have to do it regularly over space and time. So another approach is amplicon sequencing. It's also a lot cheaper, and I'm going to talk a little bit about that. So those populations that are quite different, the Benin population, the North Ghana population, we can see parts of the genome that they don't map to the reference genome. And so what we want to do now is actually go and create long-read data from these discrete populations that we're seeing and understand more about the genetic variation in that space. Okay, so you might have heard, probably not, but some of you might have, that people are very, very up in arms about this Anopheles stevensii invasion of Africa. It's a really serious malaria vector, urban vector in Asia and in India. It's very bad. And it's got into Africa. And different funders have thrown a lot of money at trying to understand how far it has spread and how much is it causing malaria outbreaks. It was discovered because malaria was nearly eliminated from Djibouti. And then in 2012, there was a big resurgence. And when they tried to figure out what species were responsible, it was shockingly Anopheles stevensii. And now it's been found across other countries in Africa. I think that there was no way to have known this because the way that we do diagnostics until recently has been to do morphology, which I told you is really difficult. You can't very easily get them down to species. And especially if the key you're using is an African key, you're not going to be thinking about, could this have been an Asian vector? And this is really important thinking about where we're going now with climate change and with malaria and the movement of the parasites and the mosquitoes outside of their normal ranges of their normal ecological ranges. So people weren't looking for stevensii, and the tools they had would have never told them that stevensii was there because they just do these diagnostics with morphology or with PCR, where you just sequence, you amplify a bit of DNA based on the primers that you use, and then you see what size it is on a gel. You don't ever sequence it. And so if you get a band that's about the right size, you just think, oh, that's Anopheles gambiae. No, you wouldn't think it was Anopheles stevensii. More often, actually, you get no band. And so that's been a real bugbear of mine, and it's influenced the panel that we've designed, because no band doesn't necessarily mean you had bad DNA. It could mean that you have the wrong target, and you're looking at a species you didn't expect. Anopheles diversity is super complex. There's over 500 named species, and they can share alleles. They're not always good species. In Africa, each of these groups, the Gambiae group and the Phonestis group, has at least 10, probably more, named species that are a mix of malaria vectors and non-vectors. They all look exactly the same. And each of these species has its own language. So you have to be working on Phonestis, and then you know exactly what tool to apply to make sure that it's Phonestis. That tool won't work to tell you it's Gambiae. So I wanted to come up with a way in which we could have a tool that was agnostic. You don't know what it is, you just know it's a mosquito. You know it's an anopheline, which is quite easy to see because they have long palps, and you want to know what is it. And the best way to do this kind of thing is sequencing. The Anopheles genus, so this one genus of one group of mosquitoes, is over 100 million years old. These are extremely diverged organisms. They are nothing like each other, even though they all look the same. There are, as I said, over 500 species, and about 10% of those transmit human malaria around the world. So this benefit of having reference genomes for lots of different species has meant that we could look at them in a comparative framework, and each of these little colorful squares is meant to signify a little place in the genome that we could design primers that were conserved across that 100 million years, but in the middle, the part that you'd amplify and sequence, was phylogenetically informative. So it would tell you, this is the species, this is the species that you're looking at. So the primer pool works on any anopheline, but the DNA that you sequence is giving you a signal of species. So we designed this primer pool, and this is one PCR reaction that happens with 62 primer pairs in it that amplifies the mosquito DNA, and 2 primer pairs in it that amplify the plasmodium DNA in the mitochondria, and that tells us what species of plasmodium is there, and also if that mosquito is carrying plasmodium or not. We call this tool ANOSP, and we think of it like a Swiss Army knife. It's a really simple tool that detects malaria parasites, that identifies the mosquito species, and if you can't place a name on it, you can still place it in the phylogeny. You can say, I don't know what this is, it looks different from everything I've ever seen before, but it's in the Gambi group. It's a tool to detect the species of malaria parasites. We've seen a lot of goat malaria and buffalo malaria in our mosquitoes, and also a lot of human malaria. And it's a tool to investigate population structure. That museum story I told you about the museum wanting us to not destroy their insects, has influenced how we do DNA extractions on everything now. So we non-destructively extract everything. We ask our partners around Africa to put mosquitoes, each of these wells has a single mosquito in it, in ethanol. They ship it back to us, and we have robots that remove the ethanol and add the magic lysis buffer C, which leaches the DNA out. And then we just use 0.1% of that DNA to run this amplicon panel, which means we have almost all of it left if we want to do whole genome sequencing. So we've been able to push, the ambition is to push 500,000 mosquitoes through this process, and to also develop the tool fully so that actually national malaria control programs are going to use it. And we've had a couple of master's students come and spend time with us, and they're taking it back to their home country's national malaria control program to implement amplicon sequencing locally. So I'm really passionate about this tool. It's very simple and it's very cost effective, and it doesn't have all these problems that the other species-based diagnostics did. This is a picture of what we've sequenced so far. We've sequenced 70,000 mosquitoes. These have all come shipped Sanger. That's not how we want to do it in the future. We want actually we're just trying to get all the pieces built so that it is something as simple as PCR. Nobody ships something off to get PCR done. We're trying to build the whole analytical system and wet lab system so that people just use it, and there's a shared analytical open database where people can drag and drop their data from their samples into it, and out pops the sort of declarations of what species are present and which ones carry malaria parasites. And we're a good long way into this process, and you can see all these different colors here. The names don't matter, but all the different colors are different species. We've got a Gates-funded project in Madagascar where we've done really dense sampling across Madagascar, and there's a huge species diversity there and a lot of malaria. This is just an example data set from the Target Malaria Group, who is one of the 3 major gene drive initiatives. So they gathered a bunch of samples from Burkina Faso, and they sent them to us for ID. They had already done their ID on them. And they did a great job. And still, there were a couple of things in here that they thought were Gambi, but 2 of them were actually Anopheles nili. You can also see that those that had no blood meal visible in their gut, 7.5% of them had parasites present still. And those with blood in their guts, because the mosquito just fed on a human and that human was infected with malaria. It doesn't necessarily mean the mosquito will transmit malaria after that, but that human had malaria. 36% of the mosquitoes that had a blood meal visible actually had plasmodium presence. So genomic approaches have been and will continue to be, I think, key in the fight against malaria. Reference genomes are super important. They're a really important data resource for many different things that I've talked about today: understanding population structure, designing gene drive, building approaches to more accurately and efficiently diagnose species. As new interventions roll out, we should be prepared to expect the unexpected. We've learned this lesson repeatedly, and we need to be doing kind of surveillance, I think, at a large scale. We need to continue to sequence both whole genomes, because that's how you identify new things happening, and targeted amplicon sequencing, where you know there's activity. We have really great understanding of the parts of the genome that are really involved in insecticide resistance. We need to keep sequencing those and understand how resistance is burgeoning. And I think we're now in the early phases of trying to modify that panel I was just talking to you about, so that we can detect all 3. There's 3 large teams across the world who are doing gene drive development and have really mature approaches. And we're developing the panel to now monitor each of those 3, because we don't know which one actually will get released, or maybe all of them will get released. And then we're going to start doing baseline understanding of what do those targets tell us in a natural population where gene drive hasn't been released yet, because you will get false positives. And so actually, and you'll see some of the resistance mutations that are out there already. Okay, do I still have a little bit of time? Yeah. Okay, that is the end of what I wanted to talk to you about mosquitoes. But I thought I would give you a little snippet into a few other things we do, both in my team and at the Sanger. We have another project called the Malaria Cell Atlas, which is also has that same kind of concept of being a genomic data resource that's important for control. So when people are developing drugs and vaccines to target malaria, to target the parasites, it's important to know how those genes are expressed throughout the life cycle. And that's been really difficult to do at a single individual level. So I was talking to you about the importance of looking at single mosquitoes rather than pools of mosquitoes. We have the same problem with malaria parasites. They're single-celled organisms. When you draw blood from a person, the way that we've sequenced these in the past is to just sequence the lot. And then you get a mixed signature of what parasites are there, genetically and transcriptomically. The Malaria Cell Atlas is an initiative in my group where we are doing single-celled, so single parasite, transcriptional sequencing. So like the human cell atlas for our body, but this is the malaria cell atlas for the Plasmodium parasites. So a quick refresher on the malaria life cycle. It's very complicated. A human is bitten by an infected mosquito, and sporozoites are released into the skin, and they migrate through the bloodstream and end up in the liver, where the parasite does one round of very large-scale replication. This is asexual replication. It's just dividing. And those burst and then enter the bloodstream. And once it's in the bloodstream, it just continues to asexually reproduce. And falciparum, which is the deadliest of the parasites, does this over 48 hours. So it invades, grows, replicates, bursts, and invades new red blood cells. And it's this part of the life cycle that causes all of the symptoms and disease, like cerebral malaria that's happening in the brain, anemia. But some of these parasites make the decision to become terminally differentiated into sexual parasites. So at some trigger will happen, and the parasites are no longer doing this asexual replication. They convert into a sexual form, and then they become male and female. And then the mosquito sucks up all these parasites. It gets lots and lots of asexuals, which just die in the gut and get digested. But if it gets both males and females, those males rapidly turn into like 8 sperm, basically. Each male does mitosis super fast. It's like the fastest cell division that's known to biology. And the males, they look like little, well, I mean, they're single-celled. They're kind of like sperm, and the females are static, they don't move. And so in the context of 5 million red blood cells in a gut, the males have to find these very rare females and mate and fertilize. And then the parasite migrates through the single-celled epithelial layer of the gut, so it busts through some cells of the gut and forms kind of a wart-like structure on the outside of the gut that then a bunch of asexual replication happens. And that's creating all those sporozoites that then burst out of that and go back to the salivary gland, and then this cycle continues. So we have done single-celled transcriptomics on all of these stages, but this was all in the lab, and we wanted to take it to the field. So this is the lab atlas, and this is what I really like about this is data. This is not just the drawing of the cycle. This is the asexual cycle here, and then becoming sexual and differentiating into males and females. So the data actually looks just like the structure of the life cycle, which is very satisfying. So we've now taken this to the field, and we've looked at single-cell data from over 50 different kids who've had malaria, and we've learned all sorts of things that I could give you a whole nother seminar on, especially about the genetic diversity of strains inside infections. We know that people carry multiple different strains, but we didn't really have such a concept of how many different strains there are and how they sometimes are related to each other, have traveled together over time, and sometimes are completely different. So likely different mosquitoes have bitten that person and brought completely different genetic diversity there. We're also running a project, completely different now, off the malaria track and onto the conservation and biodiversity genomics track called BioScan. And this is, again, large-scale sequencing, but it's just a tiny, it's like the amplicon sequencing. It's just a tiny little bit of DNA from lots of individuals. So we launched this a few years ago, and we basically get volunteers across the country who are interested, for example, in rewilding. So they've They've got an area, they're trying to bring more biodiversity to that area, and they want to monitor how they're doing. It's very difficult to do this on insects. There are 25,000 species of insects in the country. There's 6,000 species of fly. You can't morphologically identify them. So you need some quick way of actually understanding what kind of diversity do you have. So very much like the ANOS panel, we send plates out to our partners across the UK. They're running what's called a malaise trap. It looks like this. It's like a tent, and the insects fly into this panel, and they basically get funneled into a jar of ethanol where they die, and each person who's volunteering plates out the insects and then sends us the plates, and we do the whole non-destructive extraction and leach out that DNA. And then we give the data back to everybody as a biodiversity report card. So here's a couple of malaise traps out in the wild. Here's a couple of images. We take a picture from the top of every specimen. So we've done over 300,000 now have come to Sanger to understand our insect diversity in the UK. And this is a little video of the website. So if you have any colleagues who are working on insects or interested in insect diversity across the UK, this is quite a fun website now. And you can see this is where all of our partners are, and there's a list of all of our partners, a lot of National Trust and different parks. That's a picture of a malaise trap. You can register interest if you want to join. And then here's the report card. So you can interact with it. If you're a partner, you can get your own data, so you can select yourself. And you can also see the diversity of species that we found. So there's 115,000 Diptera that we've collected. So that inner circle gives you the order, and the outer circle gives you the species. So in a second here, this video will click on Hymenoptera, so all the bees, ants, and wasps. And now it's doing the same thing. It's giving you a sense of the diversity of the specimens that have come in so far in our project. And this is over time. You can see the summers when we get lots more data samples. So they trap for 24 hours every month. That's part of the volunteering. And here's the sequence of the barcode that we generate that tells us that that species is a species and it's different from other ones. It's a great little, it's a fingerprint essentially, just using the mitochondrial cytochrome oxidase 1 DNA barcode. Okay, that's basically it. I just wanted to say one more thing about Sanger. So we are just down the road, and we have 6 different departments, effectively. We call them programs, and about 30 faculty teams. I sit in the Tree of Life, and the Tree of Life has this Darwin Tree of Life project. And these are all of our partners, and we've generated reference genomes for over 1,500 species from Darwin Tree of Life, and over 2,500 as just more widely across the Sanger Tree of Life department. And what I want to emphasize about this project, which I think is a wonderful, wonderful project and really deserves, I think it's changing biology forever. And the reason that I say that is because everything I just told you about today was basically based off the reference genomes of just 1 or 2 species. And you can imagine having reference genomes for all of biodiversity, what that will enable. Genome-enabled science, I think, is incredible science, and it really does have applications more broadly to especially pests and disease, but also to our biodiversity in a changing climate. And that's it. Oh, this is the progress to date. This is just a sense of, we're really sequencing everything, and we've released over 2,500 genomes. These are my funders, the Gates Foundation, Wellcome, and MRC. Petra and my team did all the wonderful drawings, and lots of people contributed to all the data I talked about today. Thank you very much.
Thank you, Mara. That was absolutely remarkable. I had no idea so much could be done. It's astonishing. I'm sure there's some questions.
I'll start with one. The amplicon panel, I think you said that your aim is to have something that can be distributed and do the complete analysis on site. Is that going ahead? Is that successful? Because it can be very difficult to do remotely high-tech operations in some of these countries.
Yeah, I think, so we have just recently got EU funding and launched, actually it will launch in July, a project called Vector Grid that will work in 5 countries to implement amplicon sequencing and to really properly do surveillance on mosquitoes in these 5 countries. We have a long way to go, but I actually think amplicon sequencing is one of, because it is actually not big data, it's small data. It's lots of samples. And we're working in the team now to try to, because of course at Sanger, we effectively have this whole branch of the Institute that is, extremely large scale sequencing. And we actually, in my team, we don't even do any of it, right? We send our samples off and it's all robotics and high tech and extremely expensive kit. And, one robot can cost £500,000. That's not scalable and feasible. So we've been working in my team to kind of reverse engineer the panel back to a normal kind of basic molecular biology lab and find the cheap robots that actually will prevent RSI from happening, desktop kind of things that move liquid off all at once with pipette tips. And we're moving the whole panel. So everything's been on Illumina so far, but we're moving it onto Oxford Nanopore successful. So that is the MinION. There's a little, you plug it into your computer and it's actually very portable and it's working really nicely. It's still not done and dusted. I mean, like you still have to have a gaming laptop. A normal laptop doesn't really work. And the analytics that I was talking about, this kind of like drag and drop to really enable people to kind of know enough, but to not have to be computational biologists to actually do the data analysis. That is work in progress. We still have a ways to go in that. But I actually do believe amplicon sequencing is feasible in country, and it's happening on Plasmodium in lots of places now. It's just not happening on mosquitoes yet.
Okay, thank you.
So I'm intrigued by the notion that you can take one of these museum specimens and take out all the interesting bits, such as the DNA, and hand them back the shell that contains no information, and they're completely happy.
Yeah.
There's something
because it's about morphology. It's not about proteins and molecules.
It's something about librarians, I think. I mean, there's just something.
We
Yes,
took all the pages and left them with the hard pages.
You've got away with grand theft, haven't you?
Possibly. Don't
Yes.
tell them.
No.
We haven't done it to all the specimens, of course. We're
Right.
very
It was
picky what
such
we
a good story. There's a question of them.
Hello.
Yes.
May I ask why North and South Africa are relatively spared from the infection? Thank you.
So North, I think, is because it's so arid, and the mosquitoes breed in water. So their adults are out and flying, but they need water to lay eggs, and the larvae live in water. South might be more about control, effective control, and housing. I think actually, it is a very treatable, it's a totally treatable disease. And it really shouldn't be what it is. There's some crazy things about it. Like it actually takes 2 weeks for a mosquito to develop the parasites to become infectious again. You wouldn't think mosquitoes typically live 2 weeks. So if you can just block mosquitoes from entering homes, or make mosquitoes' lives a little bit shorter, they won't be able to transmit. And I think that probably in the South, there's just been more like modern housing. And so mosquitoes aren't getting inside. They also like to bite at night, although some of the control measures are causing behavioral shifts as well as resistance. So there are reports of mosquitoes actually biting earlier in the day outside and transmitting malaria because they're avoiding the nets and avoiding going indoors.
Thank you for a fascinating talk. I was really interested in that you're doing, the focus of your sequencing is on the mosquito, not the parasite. And I wondered whether looking at other samples, you found mosquitoes that don't have parasites, or indeed if those parasites can potentially infect other insects. So how close is this tie? And would it be possible to eliminate the parasite without eliminating the mosquito?
So a lot of parasite sequencing, much like what I talked about with mosquitoes, has been done. So we understand population structure of the parasites really well in Africa, actually around the world. And falciparum is globally spread, and actually it's transmitted by many, many different species of mosquitoes. So the parasite, there's probably some local adaptation where South American falciparum is more adapted to South American vectors, but South American falciparum will transmit in African vectors. So targeting the parasite is that's an important area of research on transmission blocking vaccines. So the idea is you give a person a vaccine and it doesn't protect them from getting malaria. So it's like an altruistic vaccine. You would take it and it would do something to the parasites, those sexual forms. So the asexuals are just reproducing as expected. And when they become sexual, something is bound to the sexual forms and blocks them from being able to do fertilization inside the mosquito. So that's a really active area of research, transmission blocking vaccines, which is kind of what you're talking about, kind of targeting the parasite instead of the mosquito.
Can you explain the gene drive method of attacking the mosquitoes?
Generally, yeah.
Just generally,
Yeah.
how it works.
So there's two different approaches. One is called population suppression and one is called population replacement. So the idea with suppression is that you effectively kill off, you make females sterile. So you basically are spreading something rapid. It doesn't go through Mendelian inheritance. So a female, you release mosquitoes into the wild who are CRISPR modified so that they, for example, carry the cassette in a locus. That means that when it's heterozygous, so they only have one copy from one parent, they're still fertile. But when they have babies, all their babies get that mutation, not half like normal, which is Mendelian. Everybody gets it because it's gene drive. All the eggs have that mutation in it. So then all of her offspring are also heterozygous. And that basically this sweeps through the population. And when you're homozygous, you are now infertile. So it's a clever trick to kind of get things spread and then cause a population crash. Population replacement is kind of the same concept. So it's again non-Mendelian inheritance because it's gene drive, but it's actually targeting a molecule in the mosquito that the parasite needs to be able to make it through the mosquito. So something on the gut, for example, the parasite can't get through the gut and dies in the gut. So the mosquitoes are fine. They just can't support they don't go away. You're not killing off the mosquitoes. They just can't support the parasite. Both are competing ideas and making great progress, technically great progress. I think they're both subject to strong selection. I mean, the parasite will evolve around that for sure. And the mosquitoes will for sure evolve around being suppressed, right? So this is why I'm talking about, like, we need this really active, we need to know that people believe that will happen. It's just about how do we monitor it and respond actively to it.
And the timescale for this to happen,
It's political now. So it's how long will that take?
but the time for the mosquitoes to gain resistance to these new approaches.
It can happen in the lab cages, it can happen in, I can't remember, 20 generations or something.
Scary.
But, just because a bit of resistance has evolved doesn't mean the whole thing is failing. It just means you get pockets of resistance and you need to keep doing release.
Well, it's amazing what you revealed. The one insect, it worries me a little bit, I must admit. And the vocabulary, well, it's beyond my recognition of so many different problems you face. And very interesting, the fact that you have extracted DNA from museum species, and of course many of us have been to Aigues Mortes. Historically, it was in Europe, and I'm just wondering how long it'll be before it arrives in my water butt, because will I know from the nasty bites that I do get, I seem to be bait for these creatures, how long it'll be before we actually see it. I think it's already encroached, hasn't it, into Europe, if I'm not mistaken.
Which species are you talking about?
Sorry?
Which species are you asking about? Sorry.
Yeah, malaria.
Oh, how long before malaria comes here?
Yeah. Will it return? Will it return?
Will it return?
I mean, are you?
Well, there's species of
actually looking
mosquitoes
into
here who can.
maybe rather like a pandemic.
Yeah.
Could it multiply in this particular fashion?
So there are species here that transmit malaria. The reason we don't have malaria is because people don't have it anymore. And that was because the swamps were drained, and it was all actually around this region, all around East Anglia. It was called fen ague, and you can see it in the bones of people who are buried that it was Plasmodium vivax, we think, a different species, and it was killing people then. And it's been eliminated by the sort of same way that it was eliminated in the States, by swamp draining and by insecticides and by high quality housing and by good medical care. So
this rewilding of the fens is then a bad thing, is it?
no, I don't think so. As long as we keep ourselves free. But there were a couple of outbreaks of malaria, not outbreaks, but a few cases of malaria in the States in the last couple of years, which was unthinkable, right? But it was people coming back with malaria, not knowing it. And then there's local species that can transmit. So unsuspecting people who were not traveling suddenly got malaria. So we do have to be careful with our travel because there are species ready to transmit here now.
Mortality is very countable, but isn't the burden of malaria something that is a major drag on the productivity of people cultivating, or
Hugely, yeah.
and on the lifespan of populations that are exposed?
Yeah, I think it's on the kids are continuously exposed and they have to develop immunity over time. So adults are getting malaria all the time. They carry it around asymptomatically. It's amazing. You can go to Mali, where we've worked, and like 60% of people have malaria, but they don't know it because they are asymptomatic. They've developed immunity to it. But the kids are suffering, and it does affect educational attainment too, because they're sick and they're fevered, high fevers repeatedly. So the burden is enormous. I just talked about the deaths, but you're right. The burden is 200 million people a year are infected and 500,000 are dying.
We've got an online question here.
Oh. Right. I've left all my glasses on. It's from Janet Davis.
I'll use the mic.
Right. Is there a time frame for rolling out genetic intervention or interference at scale? Question mark. I think you've sort of
Yeah,
addressed that
so
question.
I think that the ambition is within the next 5 years.
And who will pay for that? Is this Gates
Gates
Foundation?
Foundation has invested a huge amount of money
Yes.
in it, hundreds of millions, and lots of funders have. Wellcome has invested a lot of money in it.
Right. So it's sort of Trump free, is it?
I don't know how safe the foundations are. I mean, I think that there are, it's not government, so they should be somewhat.
I'm interested in any unintended consequences, because presumably mosquitoes have a purpose. Is it cross-pollination or whatever?
Yeah.
Have you thought about that? What are the
Yeah.
feelings
So,
on
I
it?
mean, personally, I don't work on gene drive. We're working on all these other approaches to monitor and understand mosquitoes. But the people who are doing the gene drive research, they have huge areas of work on that because there's lots of people concerned about that, about the impact on the ecosystem, on the impact on other species locally. To date, I mean, so one way of thinking about these these 4 species I talked about today, which are spread widely across Africa and do the vast majority, like over 90% of the malaria transmission in Africa, they're kind of like rats. So they followed people around. They're very fixated on people. They're hosts. They bite people. They go in homes. They're not the kind of, which doesn't say they should still be eliminated. It doesn't mean they should be eliminated, but it also means that they're kind of, the way that we think about pigeons and rats. They're that kind of a species for a mosquito. So they probably do some pollination, but it's very unlikely that they're doing pollination on, that they're key pollinators for any plant. There was one study, I believe from Uganda, that showed that actually there is a jumping spider that lives inside homes that does like to eat these indoor resting mosquitoes. But the idea is that there's lots of other indoor resting bugs that come inside and live in your homes. So probably it's not, again, a kind of specialist interaction. It's just there's a lot of them, and so that's what they eat.
We've had a fantastic set of questions to a really remarkable talk. It's just so interesting, and congratulations on this.
Thank you.
This incredibly important work. Thank you so much.
Thank you.